Alright, ladies and gentlemen, this is going to be the next video on mutations, and this is going to be the second video here. This is going to be Mutations Part 2, specifically looking at mutations occurring along the DNA. Let's get started on this next video. So let's go through and look at point mutation types here. We're going to have substitution mutations, and then we're also going to have insertion and deletion mutations. And what we're looking at is mutations that are going through and occurring with individual nucleotides. So substitution is a type of mutation where one base pair is replaced by a different pair. This is going to result in a couple of things here. It's going to result in a nonsense, missense, or silent mutation. This will all make sense shortly. Now, if we look at insertion and deletion, we're going to see that there is going to be an extra nucleotide added or deleted, and this is going to result in a frame shift mutation along that DNA. So again, what I want you to do is take a look at this video here, look at this flowchart, make sure you understand the flowchart and where we're going through and seeing these things. And then once we understand this flowchart, we can look at this in a little bit more detail. So let's go through and look at point mutations, and let's look at a substitution mutation. We're going to look at 3 substitution mutation types that are going to result in 3 different scenarios here. So a substitution is a type of mutation where one base pair is replaced by a different pair, and the first specific type we're looking at is a silent mutation. This is where the amino acid sequence is going to remain the same. So if we have our section of DNA here, we're going to go through, here's our long strand of DNA. It's going to consist of TAC, GCC, ACC, AAA, GCG, TGG. If we look at our mRNA, what we're going to see is there's the corresponding sequence. We can use our codon table in order to determine what the amino acid sequence would be. So if we go through and look at it and use our codon table, this is what the corresponding amino acid sequence would be. Now, in a substitution mutation, we're only going to go through and substitute out one single nucleotide here. So this is going to result from AAA to AAG. As a result, we're going to see that UUC is going to go through and occur. And what we see here is in a silent mutation, our amino acid sequence stays the same. If you look at our codon table, UUU and UUC still code for phenylalanine. And as a result of that, what we see is in a silent mutation, a mutation occurs along one individual nucleotide, one of them substituted, and the amino acid stays the same. Now, if we look at a different substitution mutation, let's go through back to our original DNA and mRNA sequence here. And let's go through and change out from AAA to CAA. This is going to change us out to GUU, and GUU is going to cause an amino acid sequence to change. So we went from a phenylalanine to a valine. Now, when this occurs, when we have a substitution where one single amino acid is changed, we have a missense mutation. This is where the single amino acid is changed, and this is going to have a small effect on that protein or polypeptide sequence there. So it's important for us to understand, silent mutation, the amino acid sequence doesn't change. Now we went through, and in some cases, when we have a substitution, what occurs is a missense mutation. And sometimes that is where a single amino acid sequence is going to change. And when that amino acid sequence changes, now we can see the entire protein sequence. One individual amino acid is going to change up that sequence. Now, the last type of substitution mutation that's going to occur is when we see a stop codon added. So we went from our original sequence of ACC to ATC. And what occurs is when this new mRNA sequence AUG is added, it's going to add a stop codon that cuts the amino acid sequence short. When the amino acid sequence is cut short, we call this a nonsense mutation. And this is where a stop codon is added early and the amino acid sequence is cut short. So if we go back to our flowchart here, remember, a substitution mutation is a type of mutation where one base pair is replaced by a different base pair. We have nonsense, silent, and missense, and it depends on what our amino acid sequence is and the change that goes through and occurs there. Alright, so now let's go through and look at point mutations here where we have an insertion or deletion. An insertion is where a nucleotide is adding, causing a shift in the entire sequence. So if we insert this T into our DNA sequence, that is going to cause a shift along the entire DNA sequence there. When that shift occurs, it's going to change up all the subsequent mRNA codons along the sequence. As a result, what we see is an entirely new sequence where that individual nucleotide was added. We call this a frame shift because when a frame shift occurs, it shifts all of the nucleotide sequences either to the right or to the left. And what's going to occur is we see an entirely new amino acid sequence when that frame shift occurs. So frame shift is where the entire sequence is shifted one nucleotide over. Now in a deletion, this is where a nucleotide is removed, but it still has the same effect. When a nucleotide is removed, we're going to see all of them shift over again. The entire DNA sequence is also going to be changed, which means our codons are changed, which means that the entire amino acid sequence downstream is changed. So again, what we're getting with insertion and deletion mutations is a frame shift where everything either moves to the left or moves to the right. So again, let's make sure we understand these flowcharts here to understand the mutations. Substitutions is what we were looking at before, is a type of mutation where one base pair is replaced by a different base pair. It's going to result in 3 scenarios. Those 3 scenarios are nonsense. Nonsense is where a stop codon is added early, cutting the sequence short. We have a missense, where we see an individual amino acid sequence change. When that individual amino acid sequence changes, we're going to see a small change in the protein structure. And then lastly here we have a silent mutation. That's where when we see this, there is no amino acid sequence change because there's multiple codons that code for the same amino acid. Now in insertion and deletion mutations, this is where one extra nucleotide is added or an extra nucleotide is deleted. And this is going to result in a frame shift mutation. So what we see is the entire sequence is going to shift either to the left or to the right. When it shifts to the left or to the right, what it's going to do is cause a new amino acid sequence to go through and emerge. Now again, I realize this was a lot of information, but I would again encourage you to go through and look at this flowchart here because this flowchart will go through and really give you a conceptual understanding of the point mutation types that we're going through and looking at. Alright, so did you learn? Well, did you learn a couple of things here? Did you learn about molecular mutations, these point mutation types? Did you learn about the types of DNA mutations and the results that it will go through and occur when we see insertion, deletion, or substitution mutations? This is going to be the end of the video, but we'll see you all in class tomorrow.